we can talk about physiology, and specifically with the small intestine, it's all about absorption. So we're going to go over how different macromolecules get absorbed in our GI tract. So let's start with carbohydrates. So this is a simple carbohydrate, a simple monomer. It has 6 carbons. Here's our first carbon, C2, 3, 4, 5, and C6 is the one attached up there. And what I want to point out is that there's 2 different ways that this molecule can bind to another monomer, okay? There could be a 1,4 linkage or a 1,6 linkage. So 1,4 linkages, our body can hydrolyze these. So this would be a 1,4 linkage. If we had another monomer here, they can combine right here, and now we have a 1,4 linkage, and another one, and they're connected. The good news is our body can hydrolyze these. So if we find starch that exists in this form, we can absorb these, break them down and absorb them as monomers. 1,6 linkages are more difficult for our bodies to hydrolyze. So this would be an example of a 1,6 linkage, and you can imagine starch would exist as a combination of 1,4 and 1,6 linkages. So how carbohydrate absorption works is that first we consume starch. In our mouth, we can break it down in 2 ways. First off, chewing can mechanically break down the starch to some extent. And we also have alpha amylase in our salivary that is produced by a salivary glands that can hydrolyze some 1,4 bonds. So on the right side, I'll show you what that looks like. We consume starch, salivary amylase comes in, and it can break down some of these 1,4 bonds. Notice it can't break down the 1,6 bonds on the right. But this will ultimately yield some oligosaccharides and some polysaccharides. So it has some digestion, but not quite enough. Then this bolus of food will move to the stomach. Those amylases that existed in our oral cavity get broken down, and then the food continues to move on to the intestine. And in the intestine, we have pancreatic amylases that are released, and these can help finish the job to some extent, so they can also break down 1,4 bonds. And you end up with a mix of monosaccharides and disaccharides, some oligosaccharides, and you also end up with some of these things. I didn't point it out here, but you do end up with some compounds that we can't digest on our own. But the things that are important from an absorption standpoint are these mono and disaccharides, because these disaccharides can be broken down by some brush border enzymes. And then finally, once they're broken down into 2 monosaccharides, we can absorb those. So let's talk about some of the disaccaridases, or the enzymes that can break disaccharides into monosaccharides. If these are enterocytes on our small intestine, we have some sugars that are coming in, and the disaccharides can't freely go in, but we have enzymes called disaccaridases that can break these disaccharides into monosaccharides. So first off, we have sucrase, which can break sucrose into glucose and fructose. We have lactase, which breaks down lactose into glucose and galactose. And another important enzyme is maltase, which can break down a maltose disaccharide into 2 glucose monosaccharides. And you've probably heard about lactose deficiency, and that's because this lactase enzyme is damaged, and we talk about that later on. Let's go over exactly how monosaccharides are able to be absorbed into enterocytes. So if this is our lumen on the top, we have an enterocyte, and we have our bloodstream on the bottom. What's going to happen is that different monosaccharides can get absorbed in different areas. And I just want to point out, I wrote apical membrane here. So glucose and galactose use the SGLT1 channel to move across our apical membrane. This membrane right here that connects the lumen and the cell is our apical membrane. And then down here is our basolateral membrane, just for future reference. You can remember that the first thing they cross starts with an A, and the second thing they're going to cross starts with a B, the basolateral. And so if we have some glucose and galactose here, they can use this SGLT1 channel to enter the cell. And what does SGLT1 stand for? That is our sodium glucose linked transporter. It's easy to remember that it's a sodium. You can see that for every monosaccharide that enters the cell, every glucose, we have a sodium. So there's a sodium and a glucose linked. And what's important about this is when they test you on oral rehydration therapy, imagine somebody just got sick and you need to give them some hydration. Something you want to consider when you're doing that is you want to not just front load them with sugary beverages. You need to give them an equal amount of sodium and glucose when rehydrating. The reason being, if you give them too much glucose, then you're going to have too much glucose left over in the lumen because you need a good balance of sodium and glucose to enter the cell, and you're going to end up getting an osmotic diarrhea. Another thing I want to point out is that if we continued this process forever, what would happen is that our cells would just be full of sodium and there would be no concentration gradient to make this absorption happen in the first place. So we need to have some sort of active transport to facilitate this process. And that's done by way of our sodium-potassium pump, which pumps 2 potassiums in for every 3 sodiums out. And by doing this, we can clear up all these sodiums and get them out of the cell. And now you've maintained a concentration gradient to allow this diffusion, this co-transport of the glucose and the sodium into the cell for absorption. So glucose and galactose follow this pathway. Fructose follows a little bit of a different pathway. Remember, fructose is kind of our pentagon home base shaped monomer that has 5 sides. So I always remember fructose is 5. And that's a good way to remember that you have a GLUT5 transporter that brings fructose in. Now, once they're in the cell, it doesn't matter if it's fructose, glucose, or galactose, they all enter the bloodstream through the same transporter, which is our GLUT2 transporter. And I remember the 2 as they all go in together. Or you can remember it as the second channel that each of these monosaccharides have had to encounter. Something I want to talk about now is the D-Xylose absorption test. So how this test works is it's useful in determining why monosaccharides may not be absorbed in a certain situation. And I'll kind of go over what I mean. So how this test works is that xylose is a monosaccharide, and the only thing you need to digest a monosaccharide is functional intestinal mucosa. Recall from the Disaccharidases lectures, when I talked about carbohydrate absorption, in order to break down the starch into the monosaccharide required several steps, several amylases, and we needed the disaccaridases to make it into a monosaccharide. But once it is in this monosaccharide form, all you need is this channel to get in, or this channel, in order to absorb. So how this test works is that you consume D-Xylose, and then you measure urinary xylose levels. Remember, I don't know if you watched the gastric lecture when I talked about the Schilling test. It's the same concept where you consume something orally and you measure it in the urine. And if you measure it in the urine, that means it was absorbed in some capacity. So if you're able to absorb D-Xylose, what we know, since the only requirement for absorption is functional intestinal mucosa, we know that this is working just fine. And so if there is some sort of insufficiency or malabsorptive process in this patient, we know it's not because of the intestine, so we should look at other regions. It might be a pancreatic problem. Maybe the pancreas isn't producing enough amylases to facilitate absorption of other oligosaccharides and disaccharides. On the other hand, if you can't detect any D-Xylose in the urine, then D-Xylose wasn't absorbed in the first place, which means the only thing that could be damaged is your intestinal mucosa itself. And so you might see this in conditions like celiac disease, where you have blunted intestinal mucosa. Now we can move on to proteins, which is a little bit more straightforward. Proteins, if you'll consume proteins, and again, there's some mechanical breakdown by chewing foods. And in the stomach, we have our chief cells, which recall from our gastric lecture are our CHOMP cells because they produce pepsinogen and gastric lipase. So pepsinogen is released by these chief cells and they can break down proteins. Once this occurs, the food will then get transported into the small intestine, and the pancreas secretes a number of proteases into the duodenum. And then these proteases, are initially secreted as zymogens, and they become activated in the duodenum. And finally, as these are broken down into 1 to 3 unit amino acid subunits, they can be absorbed in our enterocytes. I've highlighted two concepts here. They're both related to zymogens. Zymogens are enzymes that require activation in order to properly function. So we just saw that pepsinogen is secreted by our chief cells. So here's our lower glandular layer. We have some chief cells. They secrete it into what's called a gastric pit. This can get released by the chief cells into the lumen, and it's released as pepsinogen, which can't do anything. But in the presence of acid, that pepsinogen becomes activated into pepsin, and this can start breaking down proteins. Trypsinogen is produced by pancreatic acinar cells, and these cells, again, trypsinogen is not active right away, but they can be activated into trypsin by a few things. They can be activated by enteropeptidase. They can be activated by enterokinase as well. They could also be activated by trypsin itself. And then again, so once activated, trypsin can pretty much activate anything in the world. It can activate, like I said, trypsinogen. It can activate chymotrypsinogen, procarboxypeptidase. And the thing you have to think about is if you have a deficiency of enterokinase or enteropeptidase in your duodenum, that could actually lead to protein malabsorption because your pancreatic proteins like trypsinogen can't get activated, even if they're secreted into the duodenum itself. A more relevant clinical consideration that is definitely going to be tested is pancreatitis, which is the opposite problem, where you actually have premature overactivation of trypsinogen into trypsin, and this can cause autodigestion of your pancreas. So let's talk about lipid absorption now. So I'm going to make this very simple to understand. Fatty acids are your building blocks of fat, and fatty acids are amphipathic. And I'm going to use this term quite a bit. And what that means is that they have a hydrophilic end and a hydrophobic end. So here's a fatty acid. You can have our hydrophilic end at the bottom, and then you have a hydrophobic end throughout most of the fatty acid. Triglycerides are three of these connected to a glycerol backbone. And again, you can notice triglycerides are amphipathic. They have our hydrophilic end and a hydrophobic end. And if we break down triglycerides, actually, let me talk about gastric lipase really quick. Gastric lipase was produced by our chief cells, our CHOMP cells in our gastric mucosa. And they have a minor role, whereas pancreatic lipases play the primary role in fat digestion. So again, this is what I wanted to show you, is that triglycerides can be broken down into monoglycerides and free fatty acids. So here is a monoglyceride. Again, you notice there's a hydrophilic end and a hydrophobic end. And free fatty acids, again, have a hydrophilic end and a hydrophobic end. The point I'm trying to make here is that whatever you have, whether it's a triglyceride, a monoglyceride, a fatty acid, they are all amphipathic. And if you try to digest an amphipathic molecules, you might have some trouble unless you have a secret weapon, which fortunately we do have. So if this is our proteases and they want us to digest these amphipathic molecules, the problem is because the lumen itself is very watery, the hydrophilic ends will form a ball and the lipid ends will kind of protect themselves in this lipid core. So if proteases try to come on in here to digest something, they're going to get a rude awakening when it's surrounded by this hydrophilic end that will take more time to digest. The good news is we have something called bile. And bile, as you can see from the lettering, how it's blue and red, bile is also amphipathic. And what that means is that instead of forming this cluster that protects themselves in these fatty clusters, you can actually have bile come in and help digest help essentially release the fatty acids, and I'll show this in a better picture in a second. And this allows the proteases to digest the fatty acids a little bit easier because it's in this, the lipid's been emulsified in some way. I think this is an easier picture to understand it. So we have our big fatty acid here. And remember, the hydrophilic ends are going to surround the outside, and we're going to have this lipid-filled core of hydrophobic material. And you can notice the fatty acids drawn there. And our pancreas, our proteases would love to come into this lipid core and digest all of that. But it's really tough because it's surrounded by this hydrophilic end. It would take a lot longer. They could still do it. It would just take a lot longer to digest it in this capacity. You're not having as much of the surface area of the fat dissolved at one time. But if you have bile coming in and binding to this region, it can actually break it down into several of these different smaller fatty acid molecules. And with these, now you can have proteases that surround each of these smaller molecules, and you enhance lipid absorption or digestion. So once the fatty acids are broken down enough, they turn into something called micelles. And these micelles or myceles are small enough to diffuse across the enterocyte membrane. Now, once they're across the membrane, they can't diffuse across the basolateral surface and into our blood vessels or our lymphatic region without a few other modifications. So the main modification required is this ApoB48 protein needs to be attached to these micelles. So you can see an apolipoprotein attached here. And once this ApoB48 protein is attached, it can be cleared for transportation into our lymphatic system. And so a mnemonic I saw in first aid is that you need ApoB48 for the B48 chylomicron lipid bombers. I don't know if you know the B48 jets that were used in wars, but this is a helpful mnemonic to remember the number 48. Something I want to point out here is that these micelles, even though they can diffuse across the enterocytes, they're still too big for the capillaries. And so instead of diffusing across the capillaries, they're actually released into our lymphatic system first. And so here's our lymphatic system. And now because it has that apolipoprotein attached to it, it can be released here. And ultimately, it will enter our systemic circulation by way of the thoracic duct into our subclavian vein. And I just wanted to point this out when we talked a little bit ago about our histology. I told you that the lamina propria has a capillary and lymphatic system and a vein. And so what's happening here is that if fat's coming in from the lumen, it's going to be absorbed into this green lymphatic system because the capillaries are a little too small to facilitate the absorption of the micelles. One thing I do want to point out is the fecal fat test. It's pretty much what you would expect it to be. It just measures the level of fat in your stool over somewhere between a 24 and 72-hour period. And a positive fecal fat test suggests fat malabsorption. And what I want to remind you, they usually don't give you a fecal fat test in isolation. Sometimes they'll give you the fecal fat test, and then they'll also give you the D-Xylose test. So imagine a fecal fat test is positive. All that tells you is that there's some sort of fat malabsorption, and it could be anything. But if they give you a fecal fat test positive, and they also tell you that the D-Xylose test shows up as positive, let's say they find urinary D-Xylose, then you know that the intestinal mucosa is working fine, and that should hone you in more. It might be something like a pancreatic insufficiency. So keep in mind that sometimes they'll give you a question that talks about both the fecal fat test and the D-Xylose absorption test to try to get you to hone in on exactly what portion of your GI tract may be injured. We touched a little bit about bile, but let's go over bile in greater detail. So bile is synthesized in the liver, and after being synthesized, it gets stored in the gallbladder. And once it's released, usually in the presence of something like cholecystokinin, it's going to be secreted into our duodenal lumen. So how that works is that we have bile, it's stored in our gallbladder, and if we have some sort of fat that's stimulating our eye cells to secrete cholecystokinin, our gallbladder will contract. And once it contracts, it's going to flow down and into our duodenum. How is it resorbed? It's not absorbed as you might expect. So the bile acids and salts don't diffuse across the membrane like the micelles did. They're actually reabsorbed in the same region as vitamin B12 is, which is the terminal ileum. And something to consider is that if you have any condition that damages your terminal ileum, not only would you have a potential B12 deficiency, but you'll end up with a bile salt deficiency. And because we just saw how important bile is in absorbing fat, this bile salt deficiency could actually give you a fat malabsorption. So this is the same picture we just showed. We have bile, we have proteases that can degrade these down into these small micelles, which can diffuse across the membrane. If the bile tries to diffuse across, it's not going to be able to. So the bile has to travel all the way down to our terminal ileum where it can be properly reabsorbed. If our terminal ileum is damaged, however, then this reabsorption isn't possible and you start losing bile. And if you start losing bile and you develop that deficiency, then you will get a fat malabsorption. The second thing I want you to know is that there are actually drugs that want this process to happen. So bile acid resins like cholestyramine, colesevelam, and colestipol, they inhibit bile acid reabsorption. And which would ultimately lead to fat malabsorption, but this would be what you'd want in a medication for somebody who has something like hyperlipidemia. And now that we've gone over both lipids and bile, I just want to mention that if you see a patient with fat malabsorption, you have to look at multiple aspects that may be contributing to the fat malabsorption. If your pancreas isn't producing enough lipases and something like pancreatic insufficiency, that could cause it. But it could also be your intestinal mucosa itself is damaged or has a problem. Your liver, if it's not synthesizing bile in the first place, that could cause fat malabsorption. And your terminal ileum, like we just mentioned, can cause fat malabsorption because there's not enough recycling of the bile into your enterohepatic circulation. Let's move on to vitamins. I want to talk about fat-soluble vitamins, then folate and B12. So fat-soluble vitamins, sometimes they're referred to as your ADEC, so vitamin A, D, E, and K. Any condition that causes fat malabsorption, you should always think about the potential for a fat-soluble vitamin deficiency as well. And the presentations are really random at times, so it's something you should always be on the lookout for. I'll give you some of the common ways these are tested. So vitamin A is found in your skin and in your visual pigments. So the two symptoms you should always look out for are skin changes. So you can see dry, scaly skin, and you can see something called Beto spots or night blindness. Vitamin D, as you might be aware of, is important for bone mineralization and calcium absorption. So again, the two things you'll expect are weak bones. So you might get osteomalacia or osteoporosis, and you might have hypocalcemia and the symptoms associated with that, like tetany. Vitamin E is lower yield, but sometimes tested. You just have to know that it protects our red blood cells. So you might end up seeing something like hemolytic anemia, acanthocytosis. You could also see demyelination of the posterior columns. I don't think I've seen a test question on that specifically. They usually talk more about the blood-related disorders. And vitamin K is a cofactor for several of our clotting factors. So I think this is the first time we're seeing the clotting cascade. All I want you to know for now is that this whole cascade, the goal of it is just to create a clot. That's why it's called the clotting cascade. I don't want to scare you about this. We'll go into this in more detail, especially in the liver section and a little bit in the pancreas section when I talk about DIC briefly. But all you need to know for now is that this whole cascade is designed to create a blood clot. And vitamin K helps to produce several of these molecules. So any factor 2, which is thrombin here, 7, 9, or 10, that can all be produced through a reaction that requires vitamin K. So if you're deficient in vitamin K, then you can't form these stable fibrin clots. And that's why the main symptom of vitamin K deficiency is a risk of bleeding. These are synthesized in the intestine, vitamin K is, and it's important to note that our bodies don't synthesize it. We actually have bacteria that synthesize most of our vitamin K. And so you could actually have in very, very young kids who don't receive a vitamin K injection because they don't receive proper prenatal care, you can get something called hemorrhagic disease of the newborn where they bleed a lot because they don't have any vitamin K stores. Vitamin B9 or folate, it's usually acquired through plants. That's you think folate is foliage. And so you don't get a deficiency in vegans. Vegans, you need to consider B12 because it's an animal product, vitamin D, and iron deficiency, but they usually don't have a folate deficiency. And you usually need jejunum and ileum to be working in order to properly absorb it. Folate deficiency is somewhat rare, though. The condition that I think they test folate deficiency on most is tropical sprue, which we discuss later. But folate, to be honest, is not as high yield. Vitamin B12 is definitely important. I went over a lot of B12 concepts in our stomach lecture, but I'll just remind us how it works. It's always good practice to remember. So our B12 is bound to animal product and it enters our oral cavity. There we have our binder produced, and each of these travel to the stomach. The acid in our stomach will cleave B12 from our animal protein, and our binder will quickly bind to B12 because if it doesn't, then B12 would get degraded by the acid. So at this time, intrinsic factors getting produced by our parietal cells, and all of this moves to the duodenum. Our binder has done its job of basically protecting the B12 in the stomach. So the pancreatic proteases will help cleave B12 from our binder. And remember the intrinsic factor that was produced in our stomach, that has also been flowing down our intestinal tract and can now bind to B12. And together, this complex can travel down to our terminal ileum. And once in the terminal ileum, this complex can be reabsorbed. And ultimately, the B12 will get stored in the liver, where you can have up to a 5-year supply of B12. I also talked about the causes of B12 deficiency. I just want to briefly go over some of the important ones. We have pernicious anemia, or if you have a gastrectomy, you might have deficient intrinsic factor production. So on this slide here, if you imagine you don't have enough intrinsic factor and we take that out, then B12 can't even get through because it needs this B12 IF complex to be reabsorbed in the terminal ileum, and you get a deficiency. For something like pancreatic insufficiency, that can cause B12 deficiency because the proteases are required to cleave that B12R binder. And if you don't have proteases, then you'll have B12 stuck to R binder. And again, that won't be able to be reabsorbed. If you have small intestinal bacterial overgrowth, what happens there is that the bacteria within the intestines actually like to consume B12 themselves. So if you don't have enough B12 binding with intrinsic factor because it's all being eaten by your bacteria overgrowth, then you won't get absorption. For Crohn's disease, what happens here is that the terminal ileum itself is damaged. So even if everything else is working properly, if you don't have a healthy terminal ileum to reabsorb this complex, then you might have B12 deficiency. And the last one I'm going to talk about is this tapeworm infection. What's kind of cool about this one is that it can cleave this complex. So it actually breaks apart this B12 intrinsic factor complex, which prevents the absorption. This I talk about in the stomach lecture about why each of these things is what it is. I just want to remind you the differences between B12 and folate deficiency and the similarities. So both of them would cause a megaloblastic macrocytic anemia, elevated homocysteine, and they could cause glossitis. But the difference is that B12 can cause neurologic symptoms and subacute combined degeneration, as well as elevated methylmalonic acid levels. And so that's how you differentiate it. I would recommend going to see the stomach lecture because I talk about why each of these things are the way they are. And again, from the stomach lecture, this is just something to keep in mind about how somebody with B12 deficiency presents. They could present with neurologic symptoms. And on laboratory findings, you're going to see not only an elevated homocysteine level, but you'll also see an elevated methylmalonic acid level. The last thing I wanted to mention is iron absorption. Usually this is hemochromatosis and is tested in the liver section, which I mentioned, and most of our section is in the liver. I just want to point out that the iron absorption is all happening in the duodenum. So how does that work? We have two different types of iron. You have heme iron and non-heme iron. Heme iron is easier to absorb. You have and it usually comes from animal products. So let's say you have a piece of steak or chicken. What's going to happen is that you've consumed some of the red blood cells, and a red blood cell is made of hemoglobins, which can be broken down into heme and globin. This heme that's in that red blood cell can be transported directly into the cell, and from there, iron can be extracted. This iron can be either stored in storage form as ferritin, or it can be transported later on. And ultimately, this iron, if it's not stored as ferritin, it can be transported into our bloodstream. Non-heme iron has a little bit different mechanism. This actually starts in this form, this Fe3+, which is called the ferric form, I should have mentioned. This cannot be absorbed so easily. You have to have it in the ferrous form, which is the Fe2+. I always remember that. I think of like the ideal amount of people on a Ferris wheel is probably 2, because you can have a nice date. And so Fe2+, the ferrous form, is preferred. But let's say your buddy Eric comes along and he wants to tag along onto your romantic Ferris wheel ride, then it becomes this ferric form, and three's kind of a crowd. So how do we absorb it? We actually need something called cytochrome B now to change the ferric form of iron into the ferrous form. And this is all done with the help of a cofactor of vitamin C. If you've ever heard that iron absorption is improved in the setting of vitamin C, this is the reason why, because it helps transform the ferric iron into the ferrous form. And now that you have the ferrous iron, it can be absorbed with our DMT1 enzyme. Something I want to point out is that this enzyme is not specific to ferrous form of iron. Anything that has a divalent metal, any of the divalent metals, which just means the 2+ metals, like all of these, can also be absorbed. So if you took iron tablets with a glass of milk, you might actually decrease your absorption because the calcium will use the DMT1 transporter and the iron might just go down your GI tract. Again, this can either be transferred into ferritin, or it can use that same ferritin enzyme to enter our bloodstream. Once it's in our bloodstream, it's often transported as this ferric form, and it's bound to this protein called transferrin. And once the whole point of this ferric form is that it could be stored in a number of different body parts. So it can go into your spleen, it can go into your bones, and it could also get stored in your liver. How is this regulated? Imagine that you had a ton of this iron floating through your bloodstream. You could have a problem. The good news is our liver can produce this enzyme called hepcidin or hepcidin, and hepcidin can come and bind to these ferroportin enzymes and turn them off. And if you don't have active ferroportin, then what's going to happen is that none of the iron can actually go from your enterocyte into your bloodstream, and you'll have less iron. And so in something like hemochromatosis, the whole problem of hemochromatosis, it's not necessarily a liver problem. It's that you have defective hepcidin or hepcidin. And if you have defective hepcidin, these ferroportin channels can work just fine, and you could have a ton of iron flooding your bloodstream. Thanks for watching. I hope you enjoyed the video. Please like, comment, and subscribe for more content.